Uh, Good morning, everybody. Uh, Today, Alper will not join, and we will go through this fourth session with Arthur and I. And let me share my screen. So today's session, we will talk about RNA-Seq data processing and analysis. So basically, we will use two different aligner programs. One of them is called STAR, and the other one is called RSAM. And we will do the genomic alignment and then transcriptomic quantification. We will create BAM files by using these programs, and we will use those BAM files to visualize our reads through the genome by using IGV. And so before starting to going details, I will start the ELISA's videos. Let's start with the genomic alignment video first, and then we will talk details about it, okay? You will go through the first part of this session. So let me check over here. So we will first create a genomic and transcriptomic indexes for STAR and RSAM at start. So let me go through the steps that we're going to follow. So by the way, do you have any questions so far? And probably Elsa will join us, and Alper might join later in the session, and you might ask questions related to more specific topics you found. So before we start, we need to use our SSH command to connect our cluster through terminal. Then we need to start an interactive session. So you already know how you can start an interactive session. So this is just a short version of it. So if you just execute this line, it will start your interactive session. So if you are wondering what's inside of it, you can use the cat command and see inside of it. And now, before you start, if you don't have this RNA-Seq directory inside the bootcamp folder in your home directory, you can execute this command. It will prepare all the necessary files that you need for the session. So after you start the interactive session, you need to load 3 softwares. One of them is called STAR, second one is RSAM, and third one is SAMtools. And so first you're gonna create index files for STAR, and they will look like this. Those index files look like this. And then you're gonna create index files for RSAM. This is the command you're gonna execute. And this is the RSAM index files. And then you do the genomic alignment with STAR first. And then you can convert the SEM output to BAM over here. You can do the sorting and indexing. And then for the quantification, I will continue with another video. Then we will go to the details. So we will be on the Slack right now. So you can ask your questions through the Slack. So hello, everybody. Let me share my screen again. So basically, you should have created those index files and do the genomic alignments with the STAR and created those BAM files afterwards. And right now we will focus on RSAM. And for that one, we will watch the video for the transcriptome quantification. and let's go through with the details afterwards. So we come to this point actually. So right now we can start doing the using RSAM and do the quantification. Beforehand, do you have any questions so I can go through with it? So if you don't have any questions, so this is the parts we're gonna do. We're gonna start with the exercise 3. We do the calculation of the expression by using RSAM. So it will create these results inside these genes.results file. You will have this expected count values, TPMs and FPKM values. Also, we will have these genome BAM files that we're going to use later on. And then afterwards, you will create these consolidated tables by using RSAM generate data matrix command. You will create a consolidated matrices, and then you're going to check the results like this. So let's give it again like a 25 minutes, or let's say 30 minutes for this part, and we will meet again and we will be on Slack right now. Hi everyone, Elisa is here, so if you have any questions, please go ahead and ask her.
Hi, Lisa. I have a quick question. You mentioned that for single cell RNA-seq, it's not possible to do paired end. Is that correct? Or is it just because to get the proper depth, you don't do paired end, but is it possible at all to do paired end with a single cell RNA-seq?
Yeah, that's a good question. It is, it just depends on the protocol. So most of the common ones that are using droplet-based technologies, they are single end because the other end of the molecule reads the barcode for the cell and for the transcript. So it doesn't actually get to a useful transcript read. But for plate-based protocols, for example, SmartSeq, then they can be paired end in which both ends get the information for the actual transcript molecule.
Okay, thank you. And then I had another question. If you wanted to do, is it possible to do different size molecules in the same sequencing run? So if you want to do like microRNAs and long non-coding RNAs and then gene encoding like transcripts from genes, is it all possible to do in one run, or do you have to do different experiments for that?
Yeah, that also depends on the library construction method that you use. So for example, yes, you can have total RNA sampling, and the thing is that to enrich for useful molecules such as microRNAs and coding gene transcripts and non-coding gene transcripts that you want, you would have to deplete ribosomal RNA, for example, because if you select just total RNA from a cell, the majority of it is going to be ribosomal, and likely that's not what you want to see. So the common ways to do this are either to get rid of the ribosomal transcripts by some kind of depletion method using probes, or to select for poly-A transcripts, which would mean then you would only have poly-A transcripts and you would lose the microRNAs, for example. Another way to select for it against the things you don't want is to select for size. So you could get only microRNAs and only small molecules like that, but then you're losing the bigger things you want. So it's all a matter of the library protocols that you choose, and based on what you want to see in the end and what's the most cost-effective way to get at that, then you select the proper approach. But there's multiple ways of doing it.
Gotcha. Thank you.
No problem.
Hello.
Hi, Alper.
Oh, sorry, I had other meetings. How is everything?
Yeah, hopefully they are doing well.
Okay. Thanks,
So you
Elsa.
guys have been going through transcript quantification, the exercises. Is that what it is?
Yeah.
Hey, thank you for the workshop. So I have a question about the quantification file. So, yeah, quick. So simply my question is, so my data is bacteria data. So I use another pipeline. I use BWA and to align the sequence, to map the sequences, and then use HiSeq to generate the count matrix. But however, the the output is all zero. So I try to find what would be the reason. So far, I think maybe the GFF file I'm using, and there are something wrong. So then I'm curious about the difference between GFF and GTF. Yeah, that's just the background and my question.
Okay. So let me break it down into a few different things and you tell me if I'm going the right way. So you're saying you're using BWA for
Yeah.
mapping your transcripts, your reads against the genome of bacterial species. Is that what it is?
Yes. And the genome is little bit different. My draft assembly genome is like over 100 scaffolds. Yeah, it's not a.
Okay. It's, yeah, multiple scaffolds. It works like multiple chromosomes, so it's not really a problem. So in the end, did you see the quality of the mapping from the BWA? Did you get most of your reads mapped, or is there a problem in the alignment step already?
Oh, I just I don't know how to qualify whether they are. I just opened the SAM file. It looks fine. It looks normal.
Okay. Yeah, so I think there is a couple of, Alper, you like some of the different pipelines for quantifying how many reads mapped and how many mapped to um, regions of interest. I think RACQC, what was it that you like
No,
to use?
feature counts. So there's another one.
Yeah, feature counts.
So
Yeah, so those will help you get those numbers.
yeah, there was this question there I was trying to answer on Slack. So you can use star for genomic alignments or transcriptomic alignments, but you need a quantifier to estimate the gene or isoform counts. That quantifier can be RSAM or feature counts, or there are many other tools like that. But RSAM is actually using a likelihood function to calculate the origin of the read, and by also taking into account the fragment length. And the estimations are usually better than other programs or algorithms. But when you look at the correlation between feature count results and RSAM results, they're like almost 99, like 0.99. So they're very close. They are both doing good job, but I think according to my experience, RSAM is doing much better job if you are quantifying isoforms, for example, or some.
Yeah, this,
Yeah, do you have
yeah,
any?
I'm just saying, like in her case, this might not even be the issue because she's just doing bacterial genome.
Oh yeah.
So it might just be a question of, did your alignment work to begin with? And I think there are ways of counting how many reads mapped. And even each aligner has to give you an output file that tells you what the performance was, how many of your reads mapped, and which, how many of them mapped uniquely. And those sorts of information are the most basic that will let you know if you can move forward, because the reason you're getting zeros in a later stage could be that nothing aligned properly to begin with. So you could be having a problem in that stage. And as for GFFs and GTFs, they are slightly different formats. If you guys want to know more about each of them, you can go to UCSC They have frequently asked questions, and they detail what each type of file format is like and what information it contains. But basically, they're both transcriptome annotation formats, and they should have the information of genes and where they start and end, where the um, UTR positions are, and what are the exons and introns starts and ends. So they both contain that information. They're just slightly different structure. That's all.
Yeah, thank you.
And each program is going to accept one or the other, but some may demand that it be specifically on one. So if you input the other one, it will not work because
So,
it won't understand the fields that it contains.
so maybe I need to Google online, so how to transfer the format.
I mean,
First,
I
you
wanted
need
to
to
ask
see
you a
if you
question.
need to have a specific one, and then yes, how to convert them, or just download a different one.
So one more thing I want to add, sorry. So this feature counts is 10 times faster than RSEM. So if you, especially for bacteria, if you don't have introns, for example, you don't need RSEM. Or if you're quantifying
Or
some
isoforms, yeah.
you don't need RSEM. Or if you're quantifying something shorter or without like any like intronic regions. So you can just use feature counts. It's much faster and you don't need RSEM for those. Like, okay, yeah, you can ask now.
Amy, I wanted to ask, so your GTF file and your whole genome FASTA file, are they from the same source?
Yeah, this is a good question. So the genome, the whole genome is I assemble them. Then I just use annotation software to generate this GFF annotation file.
So you got your GTF from GFF, right?
I don't have GTF. I just have GFF.
GFF.
Yeah, generated from an annotation file, Proca or Prodiga. Yeah, all the server Rust.
Yeah, because sometimes if your GFF or GTF and your whole genome are not from the same source, like if you did like GenCode versus and some like and UCSC, they're from different sources. It's difficult to actually to annotate. So that's probably sometimes you would see zeros like that. So you should check that.
Yeah,
that's a good point.
Yeah, I think
if you have, for example, something called gene A on your GFF, and gene A, the position doesn't exist in your mapping file, then it will get 0 counts because no reads map to it. So everything needs to be compatible. That's a great point.
Yeah, I think this might be my the reason, because I also use another software like figure counts. So I use two, so both of them generate the 0 result. But yeah, but in my case, I don't know how to make sure that the input files are from the same source.
So one way would be to just check that your chromosome names match. In your case, you have scaffolds, right? So each of them will have a name in your FASTA file. Those names have to be the same names that are used on a GFF file to say where the gene starts and ends and where it's located.
Oh, okay, thank you. I will check the name of the column in the GFF and to make sure. Thank you so much.
All right.
Do you have any more questions for Elsa? Okay. Yeah.
Hi. Oh, I guess I did have a question. I just asked on Slack, but it seems like more of an open-ended thing. I guess I was wondering if we're going to talk more in this class about, we said that STAR is good for aligning things without a known transcriptome, and then RSEM is good for doing differential expression when you have a known transcriptome. Are we going to talk a little bit kind of linking those two together and essentially identifying new genes, new transcripts, and then quantifying them? Like, is it just as simple as like running STAR and then running RSEM? I imagine there's some extra step in the middle where you actually define new annotations. Is that something we're going to cover later, or if there's some way to look into that, I'd appreciate that.
I'm leaving it to you guys. I can
No,
go on it, but what we're going to cover is probably more on your end.
So,
Yeah.
okay, like we can tell you a couple of things about this. So this is a different story. Like you need to do transcriptome assembly if you want to find unknown genes or unknown isoforms for your samples. So it's a different thing. So what we are showing here in this bootcamp is usually transcript quantification or transcript, not transcript annotation. So if you have a special transcriptome, or if you did something special to your samples, that if you want to detect new isoforms, new transcripts, if you expect such things in your own like samples, so you need to first map everything with star. Star will tell you at least where your reads are mapping in whole genome plus transcriptome. Even they are not annotated, the reads will go somewhere in the genome. So the second thing, after you run star, so you're going to have an alignment file. Then there are some tools like cufflinks, for example, or there are many other tools like string tie. We are using string tie for that purpose. So what you're doing, it's doing transcriptome assembly or transcriptome detection, transcript detection, and then it will tell you the locations. And then after you run that software, you are also running other tools to annotate that detected transcripts and to find their names if they exist, or if there is a gene in that location in that transcriptome, it tells you that. So there are a couple of more steps, but the beginning is the same. Like you need to prepare, align them with star, and then run a couple of more transcriptome assembly software. So then it will also quantify those isoforms or genes, and then it will tell you if there's a name for it. The transcriptome annotation softwares will also tell you the names of those regions. But at least, but this is a different path, not the path we are showing in this bootcamp. But if you have any questions in the future, so we can work together to tell you what software we are using for this purpose.
Awesome, thanks. That really helps sort things out. Appreciate it.
Welcome.
All right. So I think we can thank to Elisa. Elisa,
That was fascinating.
are you planning to come next week? Again, for the questions.
I think, let me just quickly check before I say yes. Yes, I can come next week, maybe around 10 a.m. again. 11, I have a meeting.
Okay.
Or 11, I can come.
Okay. Thank you
So
so
just
much,
tell me
Elisa.
when you guys want. No worries.
Thank you.
Bye, everyone.
Bye. Alper, do you want to continue or do you want me to continue right now?
Oh, you should finish.
Okay. Okay. So let me share my screen now. So we finished the first two parts of our session, and lastly, we're gonna go through the visualization step. So again, I will start with the short video of Elisa's, then I will go through the details of this third session. So right now we have created a quantification file, and we have created our sorted BAM files, and we created their index files afterwards with the SWAM tools index commands, so we can load them into our IGV program. So the way that we can do is simply we need to first download the IGV. If you don't have, please go ahead and click this link and download IGV. Then you need to copy those files to your computer in order to easily visualize them. So you can use FileZilla for it, and simply in our first session we have downloaded FileZilla, and you can come to this part and enter ghbc06umassrc.org over here as a host, and your username you're going to enter and password, and then you're going to click this quick connect button, and you will connect like this, and you will see all the directory structure over here at the right part. So in the you will find bootcamp folder, then RNA-Seq, and you will have this sorted folder. So, and inside the sorted folder, you have all the BAM files that you need. You can simply drag and drop this sorted folder over here to initiate the copy. And now, after this transfer is completed, then you will have this sorted folder, which has all the BAM and the index files in the same folder. So the next step is going to be to open up a IGV and choose your genome, which is mouse MM10 for our test scenario. And then we will load our files. So we will go to that directory and we will choose the BAM files that we want to load, like this. I'm not going to choose the index files, but IGV will use this information to load BAM files into their system. So now we have loaded these files into the system. Next step is simply we can search one of the genes that we are interested in. Right now, we know that this genome is available. Let me search again. So yeah. Let me search maybe another one. This one looks much more easily. Yep. So this is the genome location, and the reads that are mapped to these regions are located over here. So the last three are all RSAM. By the way, I just loaded the RSAM results, and the last three is experiment data, and the first three is the control data. As you can see, the heights are quite different, so you can see the scales over there. So please go ahead and try these parts, and then we might talk it about later. Okay, we will be on Slack.
Quick question, please.
Of course, yeah.
So can this also be used for representing small RNA-Seq data?
Yeah, sure. So all BAM files can be loaded into the system.
Okay. Thank you.
No problem.
Maybe I can tell one more thing here about other genomes or some small RNA-seq sequences you defined, that how you can actually visualize them using IGV. So there is this, there's a section in IGV or UCSC genome browser. So what you can do is you can upload your own sequence files as a new genome. And then, for example, if you just align your reads to small RNAs, for example, and only microRNAs, maybe immature microRNAs, you just want to see where they mapped in those sequences. So those are the mature side or 5' end of the microRNAs or 3' end of that microRNA sites. So if you want to visualize them, you're just going to create a FASTA file and upload as a new genome. And then when you map your small RNAs to that small RNA FASTA file, then you can actually upload that BAM file using your own genome file. So you can define any genome files like that. It doesn't have to be like whole genome. It can be any FASTA file. But the only thing you need to do is after you define your own FASTA file, and then you need to align your reads to that FASTA file, they have to be they have to be used together. So for example, if you align your reads only transcriptome, for example, and transcriptomic mapping reads and their coordinates will start from 1 to length of the gene. So it's going to be somewhere in between those the read locations. So they're not going to be in genomic coordinates. So if you want to visualize these reads in IGV, you need to upload the transcriptome FA file there. So, but if you align them to whole genome together with transcriptome, and if you have the genomic coordinates, then you have to use the whole genome there. So you have to be careful what kind of alignment file you have, and you have to match your reads with your aligned genome or transcriptome. But when you align your reads with RSAM, you're going to have like two BAM files there. One file will be genomic, those coordinates, mapped coordinates going to be in a genomic coordinates. The other file will be in a transcriptomic coordinates. So you have to be careful there. So you cannot upload that transcriptomic, the file that has transcriptomic coordinates in it to IGV and try to visualize it, the one in, like you're doing now in mouse. So you have to understand like where you map your reads, where you align your reads, and then which genome file or transcriptome file you're going to use. Okay, if you have any question, this might sound a little bit complicated, but if you know exactly what you're doing, then you can actually visualize anything you want on IGV. It's open.
Thank you, Apa. I'm definitely going to follow up on that.
Okay.
I have transcriptomes and I want to map my small RNA. So that's the reason I'm asking that.
Yeah, you need to define your own FASTA file for your own small RNAs, and then create a genome file for that, and then upload that, and then align your reads to that genomic file, that your small RNA file you created, and then visualize them together. So, but we can talk that later too.
All right, thank you.
You're welcome.
So let me close the session. So please go ahead and try to follow the IGV step. And then please come to this differential expression analysis part and execute these commands to do a kind of a quick and dirty differential expression analysis. So you will have a simple results of few genes that are 11 genes are reported as important and differentially expressed, and you might easily go and check through the IGV those genes. So are they really different or not? You can check them. And finally, please try to do this session 4 homework, and we will meet you next week again. And you can always ask questions through the Slack.